The brain vasculature is a complex network of penetrating arterioles, capillaries, and ascending venules. These vessels are encased by mural cells such as ensheathing pericytes, which express alpha-smooth muscle actin and are found within the first 4 branches from penetrating arterioles. We use ACTA2 RCamp 1.07 transgenic mice to visualize calcium transients in these cells through the expression of the genetically encoded calcium indicator RCamp. Calcium plays a strong role in contraction of these cells and blood flow control. Capillary pericytes are found farther downstream in the vascular network and can be labeled in other transgenic models. However, we focus on ensheathing pericytes in this method. Our mice have been surgically implanted with a chronic cranial window that permits access to the brain vasculature for imaging. Here, we outline the necessary preparation steps and tail vein injection, followed by the 2-photon imaging and analytical methods needed to acquire pericyte calcium data and information about blood flow in anesthetized animals. The following items are required for a tail vein catheter injection: insulin syringes, a 15 centimeter piece of PE10 tubing, 30 gauge needles, gauze, saline, forceps, green fluorescent dextran dye, pliers, and scissors. We also have a needle.